would like to thank the organizers for inviting me to this very interesting meeting. I'll be talking today about the origins of life as a process of development of autonomous functional systems. As you know, the origin of life is placed in a very central position within the overall picture of astrobiology, and two different approaches have been used to study this process. First, bottom-up approaches, so from past to present. This is done by disciplines such as astrochemistry or geochemistry or prebiotic chemistry, of course, and then top-down approaches from now to the past, from the living to the non-living, as traditionally did by paleontology and also microbiology and molecular phylogenetics. We are interested in knowing what could happen between chemistry and biology, okay? In particular, we are interested in investigating the processes that could have been operating between these molecules and LUCA, that we can imagine as a cellular entity similar to this, but we don't know, and more precisely, between these molecules and a protocellular entity that could be able to undergo Darwinian evolution. Okay? So the key point here is to try to distinguish the features that evolvable individuals have, or life has, and the pure chemistry doesn't have. For that, it's quite interesting to remember a couple of the definitions of life. So we are referring to when we talk about life. Christian de Dief told us that life is a system that can maintain itself in a state far from equilibrium, and this is very important, and that can grow and multiply with the help of a continuous flow of energy and matter from the environment. So this is essential for life. Also, Jerry Joyce coined this short definition of life that was further adopted by the NASA Astrobiology Institute. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. And my friend and colleague, Kepa Ruiz-Mirazo, with Julie Peretó and Álvaro Moreno, said that living beings are autonomous systems with open-ended evolution capacity. Okay? So far for equilibrium, self-sustaining evolution, because evolution, as you know, is a key feature of life. Because as Dostoevsky said, nothing makes sense in biology except in the light of evolution. So when we focus on living beings, we see that all of them share a metabolic machinery that is required to gather energy and material resources from the environment and to allow the system to stay far away from the thermodynamic equilibrium, as I said before. Then a compartment that is used to keep the molecular components together and distinguish the organism itself from the environment, and of course, a genetic and template set of mechanisms that allow the organism to transmit heritable information to its offspring. So a combination of this will make possible the appearance of life, because these features confer autonomy, individuality, and history to life, which are essential processes for the living. The point now is to try to develop mechanisms that could allow producing some of the components of the metabolic machinery, compartment, and genetic and template mechanisms. And this has been the work of prebiotic chemistry for years, you know. Starting from the seminal ideas by Charles Darwin in his books and letters to colleagues, the first models were set, as Antonio Lazcano told us yesterday, by Alexander Ivanovich Koparin, and almost simultaneously by John V. Haldane in England. And 3 decades later, the first experiments come to the stage with the famous Stanley Miller experiment, and also Joan Oró, and Catalan and Spanish researcher, okay, who was able to produce the first nucleobases abiotically. From that, a huge number of researchers have been working in different aspects related to the production of the metabolic machinery, compartment, and genetic and template mechanisms. This is a not exhaustive list, of course. And at the end, we have a final picture that is quite complicated. We have lots of processes that could have been operating in the early Earth and leading to protometabolism, protocell membrane, and protogenome components of life. Okay? But it's also possible not to try to go to each of these components in a separate way, by to all of them together, because this is the way that life could have been using. This takes us to the systems perspective in biology and chemistry. This is something well known in biology, since the combination of experimental data and computational modeling has allowed further insights into the living processes. And this was leading to the so-called omics revolution in molecular biology since the decade of 90s. And then, later, systems chemistry was also developed as a research program that hopefully ascertained the roots of biological complexity. It was established in 2005 thanks to the seminal works of Tibor Ganti and Gunther von Kietrowski. They deal with complex systems and they study the dynamic processes that can be operating in the chemical world. Okay? So, prebiotic systems chemistry provides a more integrative and perhaps realistic approach towards the origins of life, because it allows higher levels of initial molecular complexity and heterogeneity, including aqueous environments, of course, different mixtures of molecules, mineral surfaces, compounds, whatever. And this could allow the transition from chemistry to life. The advantages of our traditional probiotic chemistry, that in my view they are not independent, so I agree with the question mark that Antonio Lazcano was using in the comparison between one-pot experiments and systems experiments. But this probiotic systems chemistry allows a more robust synthetic pathway production. Then, in my view, the replication first versus metabolism first controversy can be substituted by a more encompassing theory. And this integrative approach could explain the transition from complex, though still thermodynamically driven, because they are chemical systems, into protobiological and then biological ones. Okay? So, using this figure from a famous article by Ernst Satzmary, the combination of genome, metabolism, and compartment could lead to binary systems and then to ternary systems endowed with full-fledged biological capacity. So this could allow us to construct ternary complex systems, so proto-living or living systems from scratch, using prebiotically plausible biomonomers, self-assembly chemistries, and also autocatalytic mechanisms that are fundamental at this point, and of course allowing far from equilibrium conditions inside the system. Okay? What is very important here in this scheme is the coupling between replication of the genetic machinery and the reproduction of the whole system. Okay? So the components of the membrane, in this case, must be synthesized by the metabolism inside the system itself. This is, of course, very difficult to implement experimentally, but we have to work on that. Okay? And we are suggesting models that can allow us and other researchers to develop new experiments in this field. Because this is mixing now, these ideas are mixing with the bottom-up synthetic biology strategies, okay? So, how to go from the most complicated chemistry we can think of to the simplest biology? So, more or less from this to this. First, we think that chemical kinetics in collectively catalytic networks and also populations of self-replicating polymers could be reinforced by some processes, including mutual chemical transformation, recognition and control relationships between the components, self-assembly processes, collective synchronization of some of these transformations operating in the chemical world, and of course pattern formation and others. Okay? This could increase the dynamic chemical stability of the system by becoming functionally engaged. And in this context, we understand as function any distinct contribution by any part of the system that contributes to the maintenance and the stability of the system as a whole. Okay? So perhaps at this stage, biochemical functions appeared. This could allow developing evolutionary dynamics, in our view. Of course, this is a quite complex process with lots of question marks. And one of the open questions is how large and complex this chemical functional space must be to express enough phenotypic variety so that natural selection could operate on it. This is not guaranteed by a limited repertoire on initial molecules. And it's not guaranteed either by a single type of polymer, for instance, RNA wall with RNA-only world. Okay? Another issue is how could the reaction networks be kept together for long enough to become evolvable individuals. So it's important that any kind of compartment, as I told before, was operating in the system. And what kind of compartment plus metabolism mass genetics systems could overcome the thermodynamic barrier to complexification and produce the entities that initiated Darwinian evolution. Okay? So these are open questions. We have been working in a group of molecular control mechanisms that perhaps could support the development of these autonomous functional systems. Okay? Our hypothesis is that we can distinguish 4 interdependent control mechanisms that are required to assemble autonomous functional systems. Those systems being able to behave as units of selection, okay, with open-ended evolution capacities. These processes can be achieved by different molecular components, but they must be produced from a common web of interlinked reaction processes, and they must play a key role in the maintenance of such a web. So they must become functional, as I was telling before. It's very important here that the autonomy of the biological system will come from its capacity to synthesize all the required chemical components the system is needing, okay, at each stage. So, which are these 4 independent control mechanisms? First one is, of course, spatial control. Because compartments avoid the dilution of the chemical components inside the system, as you know, also allow spatiotemporal coordination of processes operating there, and more importantly, they allow far from equilibrium thermodynamics inside the system itself. It's important to have compartments, and as I told you, in order for the system to become biological, the components of the membrane of the compartment must be synthesized by the system itself. The second control mechanism would be kinetic control, of course, because catalytic species can allow the transition from a thermodynamically driven system to a kinetically driven one. And life could have started using organocatalysts or mineral surfaces and interlayers, also metal ions, short peptides, and further ribozymes, and so on. The third mechanism is energetic control, because energy transduction is essential for life and it's required for far from equilibrium chemistries. Bioenergetics through the membrane could allow the transformation from electrochemical potential of different substances to energy currencies, initially perhaps pyrophosphate, later on perhaps ATP and so on. And they allow coupling endergonic, among them synthetic processes, and exergonic processes. Otherwise, it's impossible to make synthetic pathways towards life. And the fourth control mechanism would be variability control, because it's essential that the parts of the system are coated in a kind of molecule that allows the transmission of the system itself. So this could be done by protogenetic molecules, perhaps pre-RNA walls and then RNA walls, in this complex framework, and of course, template self-replication mechanisms operating there. In our view, these 4 control mechanisms are interdependent. So we drew this tetrahedron to show how the kinetic control, variability control, energetic control, and spatial control are interlinked and should have appeared more or less at the same time to allow the production of living beings. Okay? After this, perhaps a more complex functional coupling was produced, and this could allow the increase in diversity and robustness of the functional couplings, and this is expressed in our drawing by a combined tetrahedron, something like this, a very weird, but okay. We use this let's say, graphic metaphor. In our world, this could have operating more or less this way. First, a set of processes, self-assembly, compartmentation, and template-independent oligomerization, acting on biomonomers and then bio-oligomers. Autocatalytic couplings and functional integration, as I told you, that could lead to this functional integration of the 4 control mechanisms. This could lead to the one biopolymer world, perhaps RNA world, and I am a strong supporter of the RNA world, but not RNA-only world, as I told you. This could allow the development of protometabolism, template-dependent replication, and reliable self-reproduction of protocells. Then further functional integration, as I told you, could lead to these multiple biopolymer worlds. With a phenotype-genotype decoupling that was previously stored in the same molecule, say RNA, and the appearance, thanks to the appearance of genetic code, this allowing an open-ended evolution towards life. So this could allow perhaps the transition from a chemical evolution to a biological evolution, and to the current biosphere at the latest. This could be an example, graphical example, artist's impression of one of those perhaps ribocytes that could combine the main components of life. And in this drawing you see here, you can recognize a genome made of RNA, then some ribozymes doing metabolic functions, not only ribozymes, alone, but in some cases combined with peptides of different length and complexity, but all of them abiotically produced. Also inorganic catalysts, soluble peptides doing other processes inside the cell. Perhaps low molecular weight molecules operating there. So this is a very complex scenario. As you see, this is dirty. This is not the kind of clean water we use in molecular biology experiments. Okay. Transmembrane peptides that could facilitate the movement of matter and energy across the boundary, and then the appearance of these bioenergetics that, as I told you, is essential for life, and a vesicle probably formed by fatty acids at the first stage. This could ensure this spatial control, variability control, kinetic control, and energetic control I was talking about. Okay? So this could have been one of the precursors of LUCA. In fact, we are far away from LUCA, as Sutherland wrote in this nice article this year. We are somewhere here. We are in the field of chemical necessity and in the transition to chemical contingency, as you can imagine, because of these control mechanisms. And further evolutionary innovations are required to go to LUCA. Okay? But in our opinion, we are setting a number of processes, of ideas, of control mechanisms that could allow a repertoire of initial molecules to interact, to make more complex systems, thanks to the emergence of these chemical couplings, this allowing the appearance of functionality in the system. So the further evolution of these kind of systems will lead to some evolutionary dead ends, of course, in some cases, but perhaps when the functional integration was stronger, some of the systems could resist this new bottleneck and give rise to this complex system endowed with a compartment, a metabolism, and replication that is, okay, ready to evolve towards different solutions, one of them being LUCA, and then the diversification of life in the tree of life, as we saw this morning. So this would allow the system an open-ended search for function. So thanks to this system and population robustness increase, we could perhaps understand the transition from chemical to biological evolution. Okay, this is what I wanted to tell you today. I would like to thank some friends of colleagues, in particular Kepa Ruiz Mirazo and Andrés de la Escosura, with whom I am working in this kind of models, and we are envisaging experiments to be done in this field. Also to the members of this is part of my experimental team at the Center for Astrobiology. We work in different aspects related to RNA world and in vitro evolution of molecules and so on. And other collaborators, friends and colleagues. Among them, I would like to highlight my friend Susana Manrubia, my friend Antonio Lazcano, because we have been talking about the origin of life, about these open questions for years, okay? So thank you very much for your attention.